Hi everyone, and welcome back to Neuropsychology. So we talked about a little bit in the last video. We talked about the introduction to our sensory systems. We covered sensory receptors and their receptive fields, and we also talked about how these sensory receptors transduce energy into action potentials. So in this video, we are going to dive deeper into the visual system. So first we're going to talk about some anatomical features of the eye, all the different parts of it. You can also see that in the picture right here. And this picture is a zoomed in picture of the back of the eye, which is called the retina. We're going to talk about the photoreceptors or the sensory receptors of the eye. And then we're going to talk about the two different visual pathways and also about the optic nerve, tract, and optic chiasm, as you can see right here. Okay, so we briefly talked about this picture before, but I just want to go over it again. So there's only a slice of electromagnetic energy that is visible to the human eye, and it lies within a very narrow range. So this range is from about 400 nanometers, which is the color violet or purpley color. And we can see all the way up to 700 nanometers, which is, we see that as red. So other animals have sensory receptors with different bands of energy. So I know, for example, butterfly can see UV. So definitely remember that our range is 400 to 700 nanometers and which color is on each end of this band. So the human eye has two types of photoreceptors, and these photoreceptors are the sensory receptors of the eye. So we have both rods and cones. So here on the right, you can see the rods, they're rod-shaped, and cones, they are cone-shaped. That's why they're called like that. So rods are the photoreceptors that are very sensitive to light, and we use rods mainly for night vision. So cones are better able to transduce bright light, and they are used for daytime vision. So then if you split the cones further up, you have three types of cones. So each of the cone types are maximally responsive to a different set of wavelengths. So we have one for the color red, the color blue, and the color green, and they mediate color vision. So the distribution of rods and cones is different across the retina. So cones are very tightly packed in a region called the fovea, which you can see right here. So in the fovea you have a lot of cones, however, there are no rods whatsoever there. So on the other side of the retina, so everywhere else except for the fovea, there are rods spread out, and there are some cones, but they're mainly in the fovea. And then right here you see this other little ditch, that is the blind spot, and there are no rods and cones right there because this is where the axons of all the neurons of the retina leave the eye. So definitely remember that. So again, in bright light, acuity is best when you're looking directly at things. That's because the cones are all together in the fovea. And then in dim light, acuity is best when you're looking slightly away. That's because the rods are on the periphery of the retina. Okay, let's go over that again. So here you have the blind spot, here you have the fovea. This whole part is the retina, and here you have the lens and the cornea. So light technically goes through the cornea and then through the pupil, which is just a hole, and then through the lens, and it will go to the back of the retina. So the fovea is the area where the cones are densely packed, and we are mainly using this for bright light and for color. And the peripheral retina has more rod cells, and rods are important for dim light. So if you recall from previous video, where there is a higher density of sensory receptors, there's an increased sensitivity. And the sensory receptors of the eye are the photoreceptors. Okay, so let's go over the anatomy of the eye. So when a ray of light enters the eye through the cornea, which kind of bends the light already, then it goes through the lens, which bends them even to a greater degree to focus the visual image. And then it will go upside down and backward all the way on the receptors of the back of the eye. So here you see the eye kind of cut open, and what I want you to see is that here you have the entire retina. Then here you have the optic nerve. Where the optic nerve leaves the eye is the blind spot. Right above that you have the fovea. So if you zoom in to this little part of the retina, or this little part of the retina, you can see this right here. So here you have the fovea, which is right there. And then here at the back of the retina, you see the rods and cones. And on the right, you see an electron micrograph of a retina. So technically here you have the fovea, and around that is the peripheral area of the retina. Okay, so how does this work, right? So light goes into the eye, then it hits the back of the retina. And then something very unexpected happens. So the flow of information in the retina is from the back of the retina towards the front of the retina. So first, light will be transduced by the rods or cones in the back of the retina. So light goes in, which is one. Then it will be transduced by the sensory receptors or cones and rods. And then that will send the information to the cell that are connected to them. So the sensory receptors are connected to what are called bipolar cells in front of them. And then bipolar cells will give their information, where's my mouse? Bipolar cells will give their information to the retinal ganglion cells. And then, so technically number 3 here are the endobipolar and the retinal ganglion cells. Then the axons of these ganglion cells are all bundled up and they leave the eye here, which is called the optic nerve, and then eventually it goes to the brain. So I just wanted to recap. Rods are sensitive to dim light and are important for night vision. Cones are used in daylight and are used for color vision. Cones are densely packed in the fovea, and in the fovea there are 0 rods. Rods are spread out over the periphery of the retina. Okay, so after light reaches the photoreceptors, aka the rods and cones, it goes to the bipolar cells, as we just said. And when the photoreceptors synapse on these bipolar cells, they induce graded potentials. And when the bipolar cells synapse on the ganglion cells, they induce action potentials. So this is the exact same thing I just told you, but we include graded and action potentials. So again, when the photoreceptors synapse on the bipolar cells, they induce graded potentials. And when the bipolar cells synapse on the ganglion cells, they induce action potentials. And the axons of these ganglion cells will collect in a bundle around the blind spot, or they also call the optic disc, and then leave the eye from the optic nerve. So this is a really cool picture. So here you see the rods and you see the cones, and then they connect to the bipolar cells, and bipolar cells connect to ganglion cells, and the axons of these ganglion cells bundle up and go towards where the optic nerve leaves. So they go out right here, and then it goes to the brain. And remember, the optic nerve, the blind spot is right here, and there are no photoreceptors there. So there are also other cells you can see right here. In orange you see the endocrine cells, and in green you see the horizontal cells, and they communicate horizontally and let other cells communicate with each other. And these cells are really important for the retina's processing of visual information. Okay, so now we covered the eye. How does it go into the brain? So just before entering the brain, the 2 optic nerves, aka one optic nerve from the right eye and one optic nerve from the left eye, they meet and form the optic chiasm. So the optic chiasm is located right here. So this thing is the optic nerve, here you have an optic nerve, and then here is the optic chiasm. I'm pretty sure chiasm means chi or x in Greek, so that makes sense because it looks technically like a big X where things will cross over. Okay, so here about half of each eye visual field is represented in the left hemisphere, and then the left half of each eye visual field is represented in the right hemisphere. So again, this is really confusing, so I definitely don't want you guys to get confused. So the right half of each eye's visual field is represented in the left hemisphere, and the left half of each eye's visual field is represented in the right hemisphere. So technically, just follow the colors. So here you can see the blue, so the left hemisphere gets information from both eyes, and the right hemisphere gets information from both eyes. So after crossing, the two bundles going to each hemispheres are now called optic tracts, because tracts are the ones that go in the brain or are in the brain. These optic tracts will then consist of retinal ganglion cell axons from the retina. So these axons go all the way from the retina all the way to the brain. And at some point, they will diverge to form two main pathways. So we're going to talk about these in a little bit more detail, but these pathways are called the geniculostriate pathway and a tectopulvinar pathway. So definitely look over this picture a little bit and maybe draw it out once or twice so you don't get confused. This is a really confusing picture where things cross over and where things don't cross over. Okay, so here I have a different picture. Here you have everything from the right visual field in green. Here you have everything from the left visual field in purple. So the right hemisphere has information from both eyes, but gets all the information from the left visual field. And in green, it gets all the information from both eyes, or it gets half the information from both eyes, but it gets all the information from the right visual field. So I don't want you to get confused there. So let's see. So what is the difference between an optic nerve and an optic tract, right? Because they technically sound the same. You would think that outside of the central nervous system it's a nerve, and then inside of the central nervous system it's a tract. So if you look at this picture and memorize this picture for future testing, the optic nerve contains information from one eye and both visual fields. So this one contains only the left eye information from both the left visual field and the right visual field. And then if you go to the optic tract, which is right here, the optic tract contains information from both eyes, but it only contains information from one visual field. So the optic tract directly goes to the brain, and the optic nerve is directly behind the eye before the optic chiasm. So I know those terms are super confusing, so I highly recommend drawing this out in different colors. Okay, so let's talk about these two major visual pathways. So the geniculostriate pathway is the major visual pathway, and it runs from the retina to the LGN, or the lateral geniculate nucleus of the thalamus. And then after it passes through the LGN, it goes to the primary visual cortex in the back of the brain, which we also call V1. So what I wanted to say is that a lot of sensory system, before they go to the primary cortex, they pass through the thalamus. But we'll cover that at the end, and we'll do a little recap of that at the end of the chapter. Okay, and then we have the other pathway, which is called the tectopulvinar pathway. This relays from the eye to the superior colliculus in the midbrain tectum, and then it reaches to visual areas in the temporal and parietal lobes through relays in the lateral posterior pulvinar complex of the thalamus. Okay, whoa, those were a lot of complicated words, right? Okay, so let's go over these again. So let's start off with the geniculostriate pathway. It's a difficult word. So the geniculostriate pathway is the major visual pathway, and it runs from the retina to the lateral geniculate nucleus, or I like to call it LGN because it's easier. And that nucleus is located in the thalamus, so it's a small part of the thalamus. So after it passes through the LGN, it goes to the primary visual cortex in the back of the brain, so in the occipital lobe. And we call the primary visual cortex also V1. So primary 1V visual cortex. So V1 is the primary visual cortex. So the V1 cortex is in the back of the brain and has a Retinotopic Map. So we're going to go over that in more detail in about 2 slides. Okay, so this pathway is really important for pattern, color, and motion recognition. So symptoms of damage to the geniculostriate system include impairments in pattern, color, motion perception, and also visual formagnosia, which is something that if you have that, you're unable to recognize objects. Okay, so here you have the Retinotopic Map that I was talking about. So let's first go over where the V1 or primary visual cortex is located. So here you have the brain, and V1 is located right here in the back. So here you see 2 posterior areas of the brain. Here you see the right side, here you see the left side. And above here you see the visual field. And what I wanted you guys to take note is if you see something in the middle, it will be processed towards the outside of the brain. And then if you see something towards the top of your visual field, it will be processed on the lower side of your brain. So everything in this map is technically upside down, inverted, and reversed. So for example, if you see something in the periphery on the right top, you will see that in the middle, it will be processed on the middle area on the left lower. So here I put how it kind of works. So middle, in the middle of our visual field goes towards the back of the brain. The right goes towards the left towards the right, up, down, and down, up. So if you see something like, for example, a tree would be right here. If you're looking at the treetop, which would be in 7 is in the right upper corner, and it will then be processed in the left lower corner. So our brains are confusing, right? Okay, so the other pathway is a tectopulvinar pathway. The tectopulvinar pathway relays from the eye to the superior colliculus in the midbrain tectum, and then it reaches visual areas in the temporal and parietal lobes through relays in the lateral posterior pulvinar complex of the thalamus. So this one is a little bit more complex. So the tectopulvinar pathway is important for detecting and orienting to visual stimuli. So as I said in the slide before, the superior colliculus projects to the cortex via the pulvinar nucleus. So this is actually really important because it provides information about spatial location of objects. So if you have damage to the system, you will have what they call visual ataxia. And this technically means not orienting in Greek. So here you will be unable to recognize where objects are located, and you won't be able to reach for them or orient yourself around them. Okay, so let's go over a little recap. I know this was a lot of information. I highly recommend pausing it and just reading through it, or going back and just rewatching this. So light goes into the eye, and then it hits the back of the retina first. Then from the photoreceptors, it goes to the bipolar cells and to the retinal ganglion cells. And then the axons form the optic nerve and leave the eye. Then half of the information of each eye crosses over through the optic chiasm, and then eventually they'll form 2 optic tracts. So these both will split up in 2 pathways in the brain. So the geniculostriate pathway is the major visual pathway, and it runs from the retina to the LGN in the thalamus, and then it goes to the primary visual cortex. The tectopulvinar pathway relays from the eye to the superior colliculus in the midbrain tectum, and then reaches visual areas in the temporal and parietal lobes through relays in the lateral posterior pulvinar complex in the thalamus. Okay, that was the end of this video, and I will see you guys back soon. And in the next video, we will cover audition or hearing.